everyone, and welcome to today's talk on achieving rapid and high-yielding cell line development. I'm here with Brett Verstak, Director of Cell Line Development at Abzena, and Jonathan Frampton, Vice President of Business Development at Unic. Today, we will be discussing the critical role of robust CLD strategies and advanced technology platforms, such as the AbZelectPRO, an integration of Abzena's CHO-K1 cell line with Proteonic's 2G Unic vector technology, which can enhance productivity, reduce risks, and accelerate the generation of IND-enabling data. Welcome, Brett. Welcome, Jonathan. It is great to have you both here with me today.
Thank you.
Thank you, Angela.
Yeah, so starting off, what is AbZelectPRO and how does it differ from other CLD providers?
At Abzena, we have a premier and highly competitive cell line development platform, which we've termed AbZelectPRO. This has been designed to accommodate and exceed our clients' requirements. The platform is the result of many years of innovation and fine-tuning, and it offers both the flexibility for the client, but also then allows us to accommodate a diverse amount of modalities that we work with. And all this is underpinned by about a large portfolio of analytical tests that are used to support lead clone selection. The platform itself has actually been heavily optimized in three key areas: the vector, the host cell line, and the process. For the vector, we partnered with Proteomic, and we have licensed in their 2G Unic vector technology, and this is now incorporated into our platform. This technology has been a huge benefit to our platform in that we've seen a real bonus in terms of boosting product titers, but also maintaining clonal stability. This venture started off in early 2024, and we were really quite surprised at how well this technology integrated into our platform and is now our current offering. In terms of the cell line itself, we use an unmodified CHO-K1 host cell line, and that has been optimized in a bioreactor to grow to really high densities, but also reduce doubling times. So this, coupled with the actual Proteomic 2G Unic vector technology, is how we're actually able to streamline our CLD process and get those timelines down to making RCBs in about 10 weeks. And then lastly, it's our process, and we've spent many years optimizing our process by bringing in state-of-the-art equipment to, again, streamline the cell line development process in general. And one unique thing to our platform is an instrument called a Cyto-Mine, which is a microfluidic device. And what this does is it encapsulates single cells into picodroplets, and then within that picodroplet, we're able to measure the amount of product that cell is actually making quite early on in the process. And then that population gets gated and printed into a well plate to assess for monoclonality. So all in all, these three key areas of cell line development is what is the whole of AbZelectPRO, and really gives us that leading advantage for our clients in giving high clonal titers, reduced timelines, and a sustained productivity and genetic stability.
Now, how did the partnership between Abzena and Proteonic stem AbZelectPRO?
The way that I see it is we've both Abzena and Proteonic have a very similar vision on what they want to deliver in terms of cell line development. It's very much supporting high titers as fast as possible and with high quality proteins. Proteonic comes at it from vector technology to improve at transcription level, translation level, epigenetic level, and by combining our technology with the Abzena technology has essentially helped to deliver that. And the alignment of the vision and mission of what we want to do naturally brought us together to then generate and create the AbZelectPRO platform.
Yeah, so the partnership began in early 2024, where the 2G Unic premium vector was incorporated into our platform and is now branded as AbZelectPRO. And that's our standard premium offering to all our clients at the moment. When we tried the vector for the first time, we were pleasantly surprised on how easily the 2G Unic integrated into our CLD platform. And we're also very excited by the uplift in the titers that we observed when we compared it to our legacy vector. So by leveraging that 2G Unic technology, we've really been able to boost our expression levels and generate really high producing, but more so stable cell lines, expressing, again, a wide variety of modalities from your standard IgG molecules to those more difficult to express proteins like bispecifics, fusion proteins, and other novel modalities.
Wow. So what makes the 2G Unic vector so cutting edge? Can you expand on that a bit?
So the thing with the challenge with protein expression is that there can be bottlenecks at a number of different stages, whether that's at whether it's epigenetically because you've got closed or open chromatin, whether it's at transcription level or translation level, or even at protein folding and others further downstream. The way that the Proteonic has approached that with the vector technology that we've developed is very much looking at trying to solve challenges at all of those levels. So the technology itself has elements that help to keep the chromatin open at all times, so you don't have stable expression. There's elements that will boost transcription, and there's also then elements that are enhancing translation. And it's doing that through recruiting more ribosomes to the site of translation, and also creating hairpin loops to get more stability. So it's not what's nice about technology. It's not just one hit on, say, transcription, but it's hit across the whole continuum from the DNA all the way through to the protein. And that then that essentially increases the likelihood and chances that it can make a big impact on increasing the titers. So I hope that gives a bit of a flavour. I don't know if you want to add anything, Brett, to that.
No, I covered it all there.
So what was Abzena's goal when pairing Abzena's host CHO-K1 cell line with 2G Unic?
Mm-hmm. Our goal was really to look at bringing in and partnering with some cutting edge technology to bring into our platform that would work quite well. We also want to differentiate ourselves from the other CDMOs in the market to provide a superior platform with very clear licensing terms, ease of use, and a lot of our competitors in the market ventured down a more of a transposase route. But to what Jonathan was mentioning before with the technology, it offers a very similar set of benefits to that of transposase in that we are seeing the stabilization, the genetic stabilization of the molecules using this vector technology and the high titers, and also the benefit of the timelines. The vector also offers a very fast way for us to progress and streamline through cell line development due to the nature of the elements that are incorporated into the vector.
So how does AbZelectPRO help difficult to express proteins compared to other platforms out there?
We've already observed a huge benefit utilizing the 2G Unic vector in our platform. We had a current program where we were expressing a bispecific nanobody fused to an interleukin, a very difficult to express protein. And what we found when we ran a head-to-head study with our legacy vector as a comparison purposes, we found that the protein was able to express to good levels, at least 3 times that of our legacy. And that was a real huge benefit for our client because we were able to produce more titer for them in such a short time frame.
I guess also to add that with difficult to express proteins, you have the challenge that you don't know where the bottleneck is in the expression. It could be, like I mentioned before, it could be at the transcription level, it could be at the translation level, and because 2G Unic vector technology is a multifaceted approach to increasing and enhancing titers, it could open up a bottleneck that you don't necessarily know where it is. And so combining that with the very well established and efficient CHO-K1 cell line that Abzena has created this very robust and premium platform for their clients.
Now, why should a customer consider an integrated CLD approach rather than standalone?
So yeah, at Abzena, we have many departments that can really help our clients take them all the way from drug discovery to IND filing and manufacturing. The AbZelectPRO platform, it is seamlessly integrated into all our wider services. It aligns with the manufacturing side of our company, and it provides that high productivity and scalability with every manufacturing run. The integrated approach also allows our clients to streamline their timeline to IND by, again, having it all that work performed under the kind of one umbrella. And along the way, we have, means of generating material for our clients that can help them see them through their support. This can be standalone material that can, act for a supply for conjugation and ADC studies, or perhaps non-GMP large-scale batches, which can be used for early process analytical and formulation development, as well as some in vivo studies. So it's by having it all in the one place that really does help having it streamline their process and make it a shorter time frame for their clients.
So what can you tell us about the track record of the 2G Unic? Any observations you can share?
Yeah, sure. I'll take that one bit. So, proteonic and the 2G Unic vector technology has been around for coming up to 10 years. We're working with a number of the very large pharma already. We've had regulatory success with the technology, with 20 known IND filings. We know that there's also currently about 20 ongoing IND projects going on currently, with at least 2, maybe 3 in sort of late phase 2. We also know that we've had a number of projects where the technology has been able to save and rescue commercially unviable products. So technically the protein is very has great efficacy, but due to very low titers, there wasn't a commercial viability. So by using the technology, we can rescue that. So altogether, we are very pleased with the track record we have, and I guess it's part of the credibility that brought Abzena to talk with Proteonic to start with.
Excellent. Great. Well, gentlemen, Brett, Jonathan, thank you so much for joining us today. If our viewers want to learn more or want to reach out, what would you recommend they do?
For me myself, if you find me on LinkedIn, always happy to make a connection, and we can happy to then answer any questions, or you can look at the Proteonic website. It's another good source of information.
Excellent.
Yeah, same goes for Abzena. Yes, find me on LinkedIn or go to our website.
Wonderful. All right. Well, again, thank you both for joining us. We really appreciated all your insights today, and we wish you all the best of luck going forward.
Thank you very much.
Angela.